Good morning, ma'am. My topic is major fungal diseases in selected farm crustaceans. Fungal diseases. Learning objectives: to detect and identify parasites in farm crustaceans. B. To know the symptoms, causative agents, and prevention of parasites in farm crustaceans. Introduction: fungal infections are common in the fish culture, but it's possible for shrimp to get fungal infections as well. It is unavoidable since fungal spores are everywhere in the air and water. Fungal are plant-like organisms, but unlike plants, are not capable of photosynthesis. All fungal diseases are called mycosis. Fungi are usually fought off by a healthy immune system, so we only see this in weakened or injured shrimp or just a mold. Spores attach themselves to weak size on the shrimp and break as a cottony white growth. If not treated quickly, this spores will invade any dead tissue cells in the process infect more tissue, causing a greater infection. At times, if the infection is only in the surface of the shrimp cell, a mold can get rid of the fungus. It is also by timeless chance that such a situation could rectify itself. Major fungal diseases in shrimps: larval mycosis, Laginium sp and Pseudopodium sp, black gill disease, Fusarium sp, red disease, alpha. Larval mycosis. Causative agents: Laginidium spp, Ceropidium spp, Halicturus spp. Species affected: all Penaeus species, crabs, and Selacerata. Growth sign: sudden onset mortalities in larval stages of shrimps and crabs. Crab eggs are also susceptible for mycotic infection. The commonly effective larval stage among shrimp species are the protozoal and mysis stage. Infected larvae become immobile and will settle to the bottom of the tank if aeration or circulation is interrupted. Presence of excessive micelle network visible through the exoskeleton of moribund and dead larvae. Effects on host: progressive systematic mycosis that is accompanied by lethal or no host inflammatory response can be observed. Infection is apparently lethal, accumulating mortality of 20% to 100 within 48 to 72 hours after onset of infection. Laginidium sp. In wild crustacean population, it is known an egg parasite, while in aquaculture, it affects both eggs and larvae of crustaceans. Laginidium calecnis is a marine phycomycetes parasite fungus in eggs and larvae of marine crustaceans. First report in larvae of Penaeid white shrimp Penaeus white seti. The hypnae of Laginidium calecnis are irregularly branched, separate and contain a cell wall and membrane, vacuoles, mitochondria, ribosomes, small red vesicles. The spores occur singly or in pairs. The fungi mycelium may either invade and embed itself in the tissues or alternatively replace all the muscle tissue of the infected larval P. monodon. Fungus infected untreated populations of Noophili, zoe, and mysis exhibited heavy mortalities. So are the species affecting of crustaceans. Laginidium infection in crustacean larvae. Larvae Larva of Penaeus monodon heavily instead infested with the fungus, completely replacing the body tissues of the larva and the vesicles, ready to release the zoospore. Filaments of Laginidium in the tail of Penaeus monodon larva. Zoospore in a vesicle and zoosporangial development of Halicturus sp by apical fragmentation, which in the arrow. Cerulpidum sv. Oral bell shrimp showing hypnae of cerulpidium inside abdominal appendages. Species Cerulpidum zopterum. Phytogenesis affects larvae ranging in stage from early villager to post-metamorphic juveniles up to 400 in diameter. Fungus spit throughout the soft tissues, causing them to disintegrate. The sporangia produce tubes which protrude outside the shell and release motile zoospore. In heavily infected larval cultures, over 90 of larvae can be killed within 2 days. Diagnosis for larval mycosis. Microscopic examination of affected larvae will reveal extensive, non-septate, highly branched fungal mycelia throughout the body and appendages. Specialized hypnae or discharge tubes, with or without terminal vesicles, may be present and could be the basis for identification of the causative agent. Motile zoospore may be observed being released from the discharge tubes in the case of some species. Classification of the types of organisms causing particular epizootic of larval mycosis is dependent upon the microscopic examination as follows. Laginidium zoospore are released from terminal vesicles. Leptorus, absence of terminal vesicles. Zoospore are released through prevention control. Disinfections of contaminated larvae rearing in tanks and chlorination and filtration of the incoming water can prevent outbreaks. Different antimycotic compounds have been tested in vitro. Use of calcium hypochloride 500 ppm for 24 hours in hatcheries is found to be mycocidal. Malachite green oxalate 6 to 10 ppb in prawn or shrimp hatcheries. 1 to 10 ppm formalin. Egg disinfection with 20 ppm detergent followed by rinsing before hatching. For Cerulpidum, no known treatment. Black gill disease, causative agent, Fusarium solani, species affected, all Penaris species. Fusarium sp. Fungi, and genus is Fusarium, species F. solani. Gross sign, appearance of black spots that mortalities in juvenile shrimps grown in ponds. Effects on host. Infectious usually starts on damaged tissues such as wound, gills, damage from chemical treatments or pollutants, and lesion resulting from other disease processes. Once infection is established, it is usually progressive with 30% remission rate. Lesions may also serve as route of entry for other opportunities. Pathogens. Diagnosis. Microscopic examination of wet mounds of infected tissues will reveal the presence of cano-shaped microcontagia. Infection may begin at different loci and spread slowly. Nested PCR EF1, EF2, primary pair. Nested PCR with FARA in both steps was tested but produced only smear or no product at all. Aflatoxicosis red disease. Causative agents. Aflatoxin produced by Aspergillus flavus and other Aspergillus spp. Species affected, Penaeus monodon and other Penaeus spp. Loss sign. Yellowish and eventually reddish discoloration of the shrimp body and appendages can be observed among pod culture shrimp juveniles. Affected animals become lethargic with weak swimming activity near pond dikes. Soft chilling can also be observed. Diagnosis. Affected will not survive for more than 30 seconds when collected from the feeding trays. There will be also loss of appetite. Confirmation is by chemical analysis. Effects on host. Hispatologically, necrosis in the tubule epithelium that process from proximal portion of the tubule's peripheral tubule. Tips in the hepatobronchials can be observed. Conclusion. Fungal disease arise due to bad hygiene. Keeping the organic load less in the system avoids majority of fungal disease. The disease caused by fungi are mostly secondary. Very few has been done on just shellfish fungus, or not much work has been done on the pathological effects on these fungus, and diagnosis techniques are also not well studied. Proper management strategies, and this is the reference